Assalamu alaikum everyone. In this video lecture, we will be discussing the paper 1 past papers of the biological molecules, which is the topic of AS level biology 9700. Let's start with MCQ number 1. What is the theoretical number of chemically different dipeptides that may be assembled from 12 different amino acids, or we can say 12 different types of amino acids? So guys, for such MCQs, there's a formula which is r raised to the power n, where r is the number of different amino acids available, and n is the number of amino acids in a chain. So over here they're saying that what is the theoretical number of chemically different dipeptides. So they're talking about dipeptides, and dipeptides are the chains which contain two amino acids, right? So dipeptides are the chains which contain two amino acids, right? So they are asking that how many different dipeptides can be made or assembled from 12 different types of amino acids. So guys, we have 12 different amino acids, which means R is equals to 12. And the number of amino acids in a chain is equal to 2, because this is a dipeptide. So the answer will be 12 raised to the power 2, that is 144. So the answer is C. Let's move on to MCQ number 2. What are the features of the triglyceride? So guys, if we talk about the triglyceride, you should know that triglycerides are non-polar and they are insoluble in water. So they are not polar, and triglycerides are less dense than water. All lipids are less dense than water. If we talk about higher energy value than carbohydrates, so yes, triglycerides have a higher energy value as compared to the carbohydrates. That's because triglycerides have more carbon hydrogen bonds, or we can say triglycerides have more hydrogens. So guys, whenever there are more hydrogens, so the molecule has more energy value, because in respiration these hydrogen atoms are oxidized to release energy, right? So always remember that point, that the triglycerides have more carbon hydrogen bonds and more hydrogens as compared to the carbohydrates, so they have a higher energy value, because in respiration the carbon hydrogen bonds are broken to release energy and hydrogens are oxidized to release energy, right? And triglycerides have a lower proportion of hydrogens than carbohydrates. This is wrong. Triglycerides have a higher proportion of hydrogens as compared to the carbohydrates. So the answer is C. Let's move on to MCQ number 3. Which polysaccharides are branched and which are unbranched? So guys, in our syllabus there are 4 types of polysaccharides: amylopectin, glycogen, amylose, and cellulose. So you should know that amylopectin and glycogen are branched, and amylose and cellulose are unbranched. So with the help of the O's, you can remember that the polysaccharides with O's are unbranched. So amylose and cellulose are unbranched, whereas amylopectin and glycogen are branched. So what should be the answer? Amylose is not branched, right? And amylopectin is branched, cellulose is unbranched. So the answer is B, right? Let's move on to MCQ number 4. The diagram shows a molecule which arrow labels a peptide bond. So guys, you should know that what is the peptide bond? If you want to search for the peptide bond, then search for CONH group. Always remember that whenever you find the CONH group, so the bond between carbon and nitrogen will be the peptide bond, right? So this is the peptide bond. So where do you find it? The CO NH group is over here, and the bond between carbon and nitrogen should be the peptide. So the answer is C. Let's move on to MCQ number 5. Which bonds are last to break when an enzyme is heated? So guys, you should know that enzyme is a globular protein, and obviously it has a tertiary structure, right? So if you start heating the enzyme, the bonds of the tertiary structure will break first. And if we talk about the strength of the bonds of the tertiary structure, so the strongest bond is the disulfide, right? It's last to break. And the weakest bond is the hydrophobic interactions, right? So guys, the hydrophobic interactions are first to break, and then hydrogen bonds are second to break, ionic bonds are third to break, and disulfide bonds are last to break. So the strongest bonds are the disulfide and the weakest are the hydrophobic interactions. Hydrogen bonds are stronger than hydrophobic interactions but weaker than ionic. So guys, the last bonds to break are the disulfide bonds when an enzyme is heated, because disulfide bonds are the strongest. So the answer is A. Let's move on to MCQ number 6. Food tests are carried out on 4 solutions. Which solution contains only sucrose and protein? So guys, you should know that for sucrose, we carry out a non-reducing sugar test, because sucrose is a non-reducing sugar, right? And for protein, what we do? For protein, we carry out biuret test, right? So we need to choose a solution which contains only sucrose and protein. So if we carry out the Benedict's test, it will be negative on sucrose and protein, right? Because there is no reducing sugar. If we carry out acid hydrolysis and then Benedict's test, so basically this is the non-reducing sugar test, right? So if we carry out acid hydrolysis on sucrose, right? If we carry out acid hydrolysis on sucrose, it will break down into glucose plus fructose, because sucrose will be hydrolyzed. And if then we apply or we carry out Benedict's test, then the Benedict's test will come positive, right? If we carry out iodine test, or if we add iodine in potassium iodide solution, the result will be negative because starch is absent. This is for the starch. This test is for the starch. And if we carry out the biuret test, it will be positive because proteins are present. So guys, the answer will be A. Let's move on to MCQ number 7. During the production of apple juice, enzymes are used to break down the components of the cell walls. Which carbohydrate will be produced by this hydrolysis? So guys, the component of the cell wall is cellulose, because they are talking about the apple juice, and apple belongs to the plant, right? So apple contains the plant cells which have cellulose in the cell wall. So enzymes are breaking down the cellulose or the components of the cell wall, and when the cellulose is broken down or when the cellulose is hydrolyzed, glucose is made. So the answer is C. Let's move on to MCQ number 8. What are the features of glycogen? So guys, glycogen does not contain nitrogen because glycogen is a carbohydrate. It only contains carbon, hydrogen and oxygen. Glycogen is a branch molecule. Yes, it is, because no O's. And glycogen does not have a structural role in a cell. It has the storage role. Glycogen stores glucose in animals, fungi, bacteria, etc. Right? Cellulose has a structural role and not the glycogen. So the answer will be C. What is the name of the bond joining glycerol and fatty acid in the formation of a monoglyceride? So guys, when glycerol is joined with the fatty acid, monoglyceride is formed, and this reaction is esterification, and the bond that is formed is called the ester bond. So the answer is A. MCQ number 10. Certain insects are able to stand on the surface of the ponds. Which property of water allows them to do this? So guys, water has a high surface tension. And what is meant by surface tension? That if we talk about the surface of water, so the molecules on the water surface, they attract each other very strongly due to strong hydrogen bonds. And because these water molecules attract each other very strongly, the surface is strong and tough. So if an insect is standing on the surface of the surface of the water will not break, and the insect can easily stand on the surface of the water, right? So what property of water allows them to do this? The answer is cohesion between the water molecules. So guys, cohesion is basically the attraction between similar molecules, and this is because of the attraction between the water molecules, which are actually the similar molecules, which provides water a high surface tension. And because of this high surface tension, the insects can stand on the surface of the water, and they can even slide on the surface of the water. So the answer is B. Let's move on to MCQ number 11. How many different polypeptides, each consisting of R amino acids, can be made if the number of different amino acids available is N? So guys, I told you that the formula is R raised to the power N, where R is the number of different amino acids, right? R is the number of different amino acids, and N is the number of amino acids in a chain, right? Number of amino acids in a chain. So in this case, the number of different amino acids is N, right? So in this case, R is equals to N, and number of amino acids in a chain are R. So N is equals to R. So the formula will be, if we say R raised to the power N is the formula, then the examiner in this example has switched the variables. So this will be N raised to the power R. The answer should be A, right? Because the number of different amino acids will always be in the base, and the number of amino acids in a chain will be in the power, right? So the true formula is this. However, the examiner can give you examples in which this formula can be switched, right? So always use this formula and then apply this formula to find the notation as desired by the examiner, right? Let's move on to MCQ number 12. The diagram shows two molecules of glucose. Four possible bonding positions are labeled P, Q, R, and S, and T, U, V, W. When these two molecules condense to form glycogen, where could bonds form? So guys, for glycogen, you should know that in glycogen there are two types of bonds, alpha 1,4 glycosidic bonds and alpha 1,6 glycosidic bonds. So bonds are formed between carbon number 4 and carbon number 1 and carbon number 6 and carbon number 1. So we need to identify the possible locations of the bonds, right? So if we talk about the bonds, then the bond can be formed between carbon number 1 of this glucose and carbon number 4 of the other glucose. So bond can be formed between Q and W, right? Another bond can be formed between carbon number 1 of this glucose and carbon number 4 of this glucose, right? So another bond can be formed between U and S, right? And these are what? 1,4 bonds, right? 1,4. What about 1,6? So 1,6 can be made between the carbon number 1 of this glucose, that is Q, and carbon number 6. This is carbon number 6 of the other glucose, which is T. So a bond can be formed between Q and T. So guys, this is 1,6. And a 1,6 bond can also be formed between carbon number 1 of this glucose and carbon number 6 of this glucose. So a bond can be formed between U and P or P and U. So guys, this will be 1,6. So these are the possibilities of bonds that can be formed. Please remember that a bond cannot be formed between Q and P because bonds are never made between the carbon number 1 and carbon number 6 of the same glucose, right? And similarly, a bond cannot be made between Q and S. A bond cannot be made between carbon number 1 and carbon number 4 of the same glucose. So guys, please be careful. So when these two molecules condense to form glycogen, where could bonds form? So what could be the answer? P and U. Is this in the possibility? Yes. P and V, it's not in the possibility. So A is wrong. P and U is in the possibility. Yes, it is. And Q and W, it's also in the possibility. So guys, the answer will be B. Let's move on to MCQ number 13. The diagram shows a molecule. Which test on a polymer of this molecule would give a positive result? So guys, this molecule is the amino acid, and the polymer of this molecule will be a protein. So they are asking that which test on a polymer of this molecule, which is the amino acid. So polymer of amino acid would give a positive result with which test? By adding the biuret solution, we will get the positive result, because biuret solution is used to test for proteins. MCQ number 14. Which property of water minimizes the temperature changes in cells and in organisms? The answer is specific heat capacity. So guys, you all know that water has a very high specific heat capacity, which means that a lot of energy must be gained by the water to increase its temperature, and a lot of energy must be released from the water to decrease its temperature, right? So because a lot of energy is required by the water to raise its temperature, and a lot of energy the water must release to decrease its temperature, so in water, temperature fluctuations are very rare, right? The temperature does not fluctuate drastically. There are very little temperature changes. So high specific heat capacity is the property of water that minimizes the temperature change in cells and organisms. So the temperature remains fairly constant. MCQ number 15. The graph shows the effect of pH on the structure of protein, which consists entirely of repeating residues of one amino acid. Which statement is true? So guys, when we are increasing the pH, what happens is that initially there is alpha helix, which is the secondary structure, right? And as we increase the pH, what happens is that till the pH of 5, we can say, there is alpha helix, and as we increase the pH above 5, the alpha helix is lost and we find the random coil. So it means secondary structure is lost. So which statement is true? That at pH 2 the protein has lost its secondary structure? No. At pH 2 the protein has its secondary structure. At pH 2 the protein has lost its tertiary structure. So guys, protein does not have a tertiary structure. So it will obviously not lose the tertiary structure because it does not have any. At pH 10 the protein has lost its primary structure. No guys, primary structure is the sequence of amino acid and it is not lost. There's still a random coil, still there's a polypeptide chain. At pH 10 the protein has lost its secondary structure. This is the answer, because at pH 10 we can see that there is only random coil and no alpha helix. So at pH 10 the protein has lost its secondary structure. Let's move on to MCQ number 16. Which molecule in the key is sucrose? Right. So guys, sucrose, is it a reducing sugar? No, sucrose is a non-reducing sugar. Does sucrose contains hexose sugar? Yes, because sucrose is made up of glucose and fructose, and glucose and fructose are both hexoses, right? So sucrose contains the hexose sugars. So the answer is C. Let's move on to MCQ number 17. Bonds are formed by condensation in cellulose, sucrose, and hemoglobin. What are the names of these bonds? So guys, cellulose is made by joining beta glucose molecules, which are monosaccharides. So always remember that when two monosaccharides or when a lot of monosaccharides are joined, glycosidic bonds are made. Sucrose is made by joining alpha glucose and beta fructose. So again, these are monosaccharides, and when monosaccharides join, glycosidic bonds are made. Hemoglobin is a protein which is made up of amino acids, and whenever amino acids join, peptide bonds are made. So the answer is B. MCQ number 18. What will break an ionic bond between amino acids? So guys, always remember this point, that ionic bonds are made between the side chains of amino acids during the formation of tertiary structure. And always remember this point, that ionic bonds are sensitive to pH changes. So when we change pH, ionic bonds can easily break. So the answer is D. Let's move on to MCQ number 19. Diagrams 1, 2 and 3 show the structural formulae of 3 polysaccharides. What are the names of these polysaccharides? So guys, always remember this point, that these two polysaccharides are unbranched, right? What are these? These are unbranched. So obviously the unbranched polysaccharides in our syllabus are what? Amylose and cellulose. So in cellulose, what happens is that you will see that every alternate ring is inverted. So for example, this is ring 1, this is not inverted, but ring 2, we can see it's inverted. Ring 3 is normal and ring 4 is inverted. So to identify cellulose, you can check for the inverted rings, right? So this ring is not inverted because oxygen is at the back, right? Or we can say oxygen is above. And this ring is inverted because oxygen of the ring is below or forwards, right? So whenever you see inverted rings, or whenever you see that every alternate ring is inverted, it's definitely cellulose. So obviously this will be amylose, because you can simply see that it's unbranched and there are 1,4 alpha 1,4 glycosidic bonds, right? So this is amylose and the rings are not inverted. So guys, if we talk about this polysaccharide, this is branch polysaccharide with alpha 1,4 glycosidic bonds and alpha 1,6 glycosidic bonds. This could be glycogen or amylopectin, right? So what are the names of these polysaccharides? So one is amylose, second one is cellulose, and third one is glycogen. So the answer is A. Let's move on to MCQ number 20. 4 sugar solutions were tested with the standard Biodex solution. The table shows the color of the 4 of the solutions after testing. Right. So guys, solution 1 gives a green color with Biodex. Solution 2 gives a blue color with Biodex. Solution 3 gives a brick red color with Biodex. And solution 4 gives a yellow color with Biodex. So guys, always remember that if a solution gives a blue color with Biodex solution, it means that reducing sugars are present. So solution 2, in solution 2, reducing sugar should be present. So solution 2 can be 0.5% non-reducing sugar and 1% non-reducing sugar, because non-reducing sugar tests negative with Biodex, right? And in solution 2, reducing sugars are absent. So there can be non-reducing sugars which could be present. So solution 2 could be 0.5% non-reducing sugar and 1% non-reducing sugar. Solution 1 is giving green color with Biodex. It means reducing sugars are present but in very low amounts, right? Or we can say reducing sugars are present in trace amounts. So this is true for 0.05% reducing sugar, because guys, 1% reducing sugar is very high. So this cannot be solution 1, because solution 1 gives the green color with Biodex. It means solution 1 has a very low reducing sugar concentration. Guys, if we talk about solution 3, it is giving brick red color with the Biodex solution. It means reducing sugar concentration must be very high, and this is true. 1% reducing sugar is true for solution 3. And solution 4 gives a yellow color, so it should have low reducing sugar concentration. So reducing sugar concentration is low. The answer should be A.